All the participants are here, and I don't think there is any questions so far. Okay.
Okay.
Stage is yours again.
Thank you. So in the first class, we learned about, I hope you are not very tired, but in the first class, we learned about what are the approaches for the simulations, and we made an introduction to the forward time simulation methods. In this course, we will learn about the SLiM and how do we run it and what should we know about at the very beginning of using SLiM. So this is an evolutionary simulation package and it runs a forward time simulations. The first and second versions of SLiM were able to run right Fischer models, but now in third version, you can also run some non-right Fischer models. But it is not very suggested to start with the non-right Fischer models. First, you can be very familiar with SLiM, how to run right Fischer-based models, which are more controlled compared to the non-right Fischer models. And then after becoming a bit advanced, then proceeding to the run non-right Fischer models. And the program is efficient and flexible because, as I said in the previous class, there is a trade-off between efficiency, flexibility, and the ease of the usage of the program. This is both efficient and flexible, and you can model and track the individuals, and you can track the selective sweeps in SLiM. The ease of use is coming from using a simple language for scripting the simulation. And the language is called as ADOS, which is invented for using in the SLiM. Maybe Greek friends can know better, but in the manual it says that the ADOS means that a form, essence, type, or species in Greek word. Plato also used this word in its forms. And this language is basic and very simple. It is a kind of hybrid of C and R. So if you have a kind of knowledge about R programming language, it won't be very easy to follow this. And there is another manual. At the end of the course, I can share the course material with you. You can also check from their website too. There is, in addition to the SLiM manual, there is an ADOS manual that you can also learn about the basics of the language. But it can be better first trying to SLiM. And then if you have some questions about writing some parts, then you can go to this ADOS manual. And this manual is showing you how to write the functions, how to produce the metrics, or what is the syntax of the language. You can check if you do correct or if you have any problem in your script from by using this ADOS manual. Then this is a kind of language like this. So what we do while running simulations is SLiM. You prepare a TXT file that you write a script like this. This is a very basic script that defines a simulation scenario. Here, what you do is talking to program to say that, hi SLiM, let's start the mutation rate as this, and let's give mutation type with a selection coefficient with X, and let's simulate to me a genomic element which is in length of 10 kb, for example. And I would like to have a recombination rate of this. Then start my population here, do something here, and then at the end of the 10,000 generation, give me an output. So you are just talking to SLiM. So if you think in that way, it can be easy to write this. And it has a structure. In all simulations, you start with an initialize part. In that initialize part, you write initialize, open and close the parentheses, and then open and close the curly parentheses, write your initializing comments to the SLiM. So this is the ADOS language. And the indentation is not very important. And after initializing the simulation, what you do is doing whatever you want and then getting the output. So this is that simple. You just need to know about how to, for example, if you would like to define the mutation rate, how to define the mutation rate. You will just initialize mutation rate. You will write this, and you can learn about all these things from the manual. If we don't cover all the subjects in the course, you can just go to the manual and incorporate more patterns to your simulation. But here, before writing the simulation, what we need to know is that, so here you write, for example, mutation rate, mutation type, genomic element type. So we need to know about what we are simulating in SLiM and what parameters do we need. So in the basic format, you're doing extended Wright-Fischer model simulation. So it is the simplest population genetic model with selection. What you do is simulating discrete generations, generation 1, 2, 3, 4, and then each generation you have individuals. And in the Wright-Fischer model, the population size is constant, but in SLiM, you can change the population size in generations. And you, in the Wright-Fischer model, again, you assume there is random meeting, and the parents are drawn from the previous generation, with the probability or proportional to their fitness. So if the parent's fitness is high, then it is selected as the parent. And then you produce the gametes by recombination and mutation. This is like this. This is from the paper of the SLiM. For example, here you imagine that you simulated one population, both copies of the genomes, 2 copies in each individual, and then there is recombination. Like the red one is recombined with the blue one. And then this is the subpopulation 2 after generating the gametes by recombination. And then you add the mutations, like the X signal here. So you add the mutations in another generation. And when there is migration, for example, from subpopulation 2 to subpopulation 1 in the other generation, draw the parents from the corresponding subpopulation. With this approach, you can simulate complex patterns of demography and population substructure and different genomic element types. Here, what does it mean? It means that you can simulate an entire chromosome with like exons, introns, UTRs, non-coding regions. So the genomic element types are those types like exons, introns, UTRs, and the non-coding parts of the genome. So you can simulate all of them. And this figure in the right side of the panel, we see the efficiency. So it is complex, but it is also efficient compared to, for example, SFS code. So the SFS code is blue and the SLiM is red. In this panel, we see the runtime. When you compare the runtime based on the locus, for example, if you, for example, sequence 1 megabase length locus, 10 megabase, 100 megabase. And here you see that the runtime for SLiM is faster. And also here, if you check here that the population size and like 200 individuals, 500 individuals, and 1,000 individuals, then again, SLiM is more efficient in simulating faster in more individuals. And then the mutation rate, if you increase the mutation rate, then introduce more mutations, but the runtime is again faster. And then the recombination rate. And in the bottom panel, you see the memory need. This is also very nice compared to SFS code. For example, in SFS code, you need almost 1,000 megabyte memory for simulating 100 megabase pairs, but very like maybe 1 in 10 proportion memory you need while using the SLiM. So again, if you simulate the population size, even if the individual number increases, the memory that is needed in SLiM is less compared to the SFS code, again in the mutation rate and the recombination rate. We see the efficiency is high using SLiM, so you can just go for it. And when we say mutations, while running simulations, while writing your simulation code, you will need to write about the mutations and the genomic elements. And the mutation types, what kind of mutations can you introduce to your simulation? They can be lethal mutations, adaptive mutations, or synonymous mutations that change anything. And to specify the mutation type in your simulation here, if you come back here, so here you specify the mutation type. You say that generate me a type of mutation which is called M1. And then here you give if it will be fixed or not. And then sorry. And then you define the distribution of fitness effect and dominance coefficient. So for specifying the mutation, you need the distribution of fitness effect, which means that this is from the population genetics courses you can remember. This means that the mutation can be beneficial or damaging or a neutral mutation. And then you need to know about the dominance coefficient. And this distribution of fitness effect varies between species and varies between coding and non-coding sequences. There is a very nice review in Nature, maybe Nature Reviews Genetics, about distribution of fitness effect. Maybe we can also check together, or maybe later. If you're interested about reading about this more, you can just write me and I can send you the paper. And different genomic element types, as I said, you can simulate. Instead of just single nucleotide sequence, like 100 nucleotide long sequence, you can just introduce UTRs, introns, and exons, and you can organize your chromosomes. So you can set a chromosome organization. You can define the location of your genomic elements. You can simulate an entire chromosome in your simulation. The basic concepts I will going to tell you about simulation before running SLiM. Before writing your code, you need to understand some basics of the SLiM. So the first thing is that what you are simulating the individuals and genomes. And this is from the manual. Here, the squares, the yellow squares are individuals. Here we have individual 1, individual 2, and individual 3. And then each individual has 2 different copies of the genome, starting from the length 0 to L. So length is L. Each square, small square, corresponds to 1 position in your genome. So each individual has diploid genomes here. And the thing is that normally our brain is very used to think about, like, we have a genome. For example, in chromosome 1, position 100, I have a nucleotide A. And then when there is mutation, it will be converted to T. So we are used to think about that there is a base nucleotide, the wild type nucleotide in our genomes. But in SLiM, you first need to learn about thinking like the genome in SLiM starts as empty genome, like without any mutations, without any genetic data. So this is called as the base version, and this is the wild type version. So you can think that base that doesn't have any nucleotides here can be neutral in terms of fitness because there isn't any nucleotide, there isn't any variation, so it doesn't affect fitness, but it doesn't necessarily to be the neutral. And if you need to start with an initial mutation state, for example, if you would like to start with a state that there is mutation selection balance, you have neutral mutations in the genome and some deleterious mutations, beneficial mutations, you can add those mutations to the particular genomes, or you can load the mutational state from backward simulator. First, you run a MS, for example, or you can use a real VCF file, real genetic data, and then feed the program with this data, then reach to a mutational state, and then start your simulations. Or if you don't have a file, or if you don't have a backward simulator produced data, then you can just run a burn-in period, which means that you don't produce the output, but run the simulation until coming to a certain generation, then generate an equilibrium level of genetic diversity through mutation selection migration balance before your real simulation starts. This is important, so you don't need to think that there are nucleotides or anything. So this is just an empty space in each individual's genome. And what we do in SLiM is like, so the adding mutations and also substitutions. And we need to learn about principles behind adding mutations to the simulation in SLiM. So for adding mutations, for example, all these three individuals were in an empty state, but to add the mutations, we need to have a position. So to which position, like 0, 1, 2, 3, to the third position, I will going to add a mutation, but to add the mutation, I need to know about its selection coefficient. And also we need to know about the fitness effect of the mutation. So if the mutation is affecting the fecundity, or if the mutation is affecting the success of the population to live. And to that, how do we know it? It is like we need to know about the dominance coefficient for the mutation. The dominance coefficient is, for homozygotes, it is like 1 plus selection coefficient. For a heterozygous genotype, it is like 1 plus selection coefficient multiples dominance coefficient. And here, how do we add the mutations in SLiM? You can, the random mutations can occur in every position based on the mutation rate that you set. If you go back, you can see here is that you set a mutation rate in your simulation. Then based on this mutation rate, randomly, the mutations will be added to the genomes of the individuals that will be simulated. Or what you can do is that requesting SLiM to add new mutation using this language, or you can say add new drawn mutation. You can read about it in the manual more. But in this case, so random mutations occur, but if you say add new mutation, then it creates new mutation, even if the same mutation exists in the same position. And also there is one idea is that, so this is an empty state, you added mutations, like imagine that here, these are not nucleotides, these are just mutations. Here, you are not simulating nucleotides. You need to first think about like simulating an empty thing, and then you are adding the mutations, just blue and red. These are not A, C, G, or T's. So here, for example, here we have blue mutation, red mutation, and green mutation. For this individual, we can say that the individual one is homozygous for the blue mutation. Normally, if you would think about like normal human genome, and if you could think that this is A and this is T, you would say that the genome is heterozygous for that position, but not. You should, the correct expression is that the individual 2 is heterozygous for red mutation and heterozygous for blue mutation. So the basic principle is that. And individual 3 is heterozygous for the blue mutation. And when you check this, you can see that genomes with blue mutation have the same shared blue mutation object. So this is just a mutation object, and you define it here like this. So the M1 is the mutation object, and you add it to your genome, not to nucleotide, just a mutation. Here, the thing is that you can think blue and red can be same mutations, but not, because they appeared due to different mutational events. So one can be introduced by the simple mutation rate, and the other can be introduced by add new mutation object. And if the 2 genomes contain the same mutation, we say that it is homozygous. But since the mutational events that generates this red and blue mutation are different, then this is not same as saying that heterozygous position. So this is not same as saying that this position is heterozygous, because individual 2 is heterozygous for red and heterozygous for blue. Then this is different. For example, in this individual, it has blue, then the fitness effect of blue is 1 plus dominance coefficient multiple selection coefficient. But in the case of the red plus blue mutation, then the fitness effect is changing. So SLiM, what does it make is tracking the mutational lineages. So if you introduce a mutation to a position, and then SLiM will track the lineage. So these 2 mutations are coming from different mutational events. This kind of difference, like having different mutations in the same positions, is fine if the mutations are neutral. So if they don't have any effect on the fitness. But in some cases, for example, if there is a soft sweep with recurrent de novo mutations, this is important. You might need to enforce the same mutational lineage every time to your simulation. So you should be aware about the distinction between these 2. And to ensure about introducing new mutations, so for example, if there is a mutation here blue, but you would like to introduce the same blue mutation, then you can use add mutations method to add already existing mutations to your simulation. While you start writing your own simulation codes, you will probably learn more about how to use all these methods in your writing. But the general idea is that you need to be sure about adding the same mutation. And another concept that we should be aware about SLiM is that what happens when the mutation is fixed in SLiM. When we say fixation, what do we say is that the population frequency of the mutation reaches to 1. So, for example, here, there is the generation first. Let's say there is generation 1 and generation 2. In generation 1, assume that randomly each individual selected one parent, and then in generation 2, we see this scenario. The blue mutation becomes fixed because the frequency, the population frequency is 1. And then if the population frequency reaches to the fixation level, if the mutation becomes fixed in all individuals, then this mutation is detected by SLiM and it is turned into the substitution state. So they are removed from the simulation. They are not added again to the other individuals, but they are also kept as substitution, which doesn't affect your fitness computations. And this is important in some cases, for example, if you study the epistasis or additive mutational effects, but not preferred. For example, if you study the direct fitness effect of the mutations, you don't want to lose the mutation as substitution, then you can just turn off this default behavior of SLiM using convert to substitution method. So if you use this method, then you can turn off the default behavior of SLiM in which it removes the fixed mutation from the simulation. If you have an idea about the basic behavior of the program, then you can just go to if you don't remember how to turn off the conversion to substitution method, then you can just go to the menu and search for it and you can find it. But the idea is that you need to know about what to do in each scenario. And also there is another concept which is not very similar to our normal knowledge about the genomes. The concept of genotype, it is a bit different in the simulation program. Normally, the concept of genotype, for example, here what we see is that wild type and blue and wild type and green mutations, we see that. But since we say that the same mutation can be added to the genome of the individual, there was a blue mutation, but the red mutation can be added to the mutation to the blue mutation too. Then the genotype concept is a bit different. Then you can say that this individual is heterozygous for wild type, heterozygous for red, and heterozygous for blue. But this stacking behavior, if you are simulating real genomes, like real A, C, T, and Gs, real nucleotides, then this stacking is nothing that you prefer because it doesn't mean anything. You don't stack A to T. But if you think that this is just a mutation, then you can stack the red and blue. It depends on your purpose of the simulation. And normally, since it stacks the mutations on top of each other while running the simulations based on the nucleotides, then you need to modify the default mutation stacking behavior of the SLiM. You can control it by using different methods like mutation stack group, stack policy, or mutation type. You can control this behavior and you can prevent stacking if you are simulating A, C, G, and T. This is also another thing that you need to consider before simulating the real sequences. Another thing is that what you do in SLiM is simulating the genomic elements. And the genomic elements can be exons, can be introns, non-coding parts, and each element can be affected by a special type of mutation. For example, if you introduce a mutation to a non-coding part, then your mutation type can be neutral. Or if you are introducing a mutation to the intron, then your mutation can be neutral or deleterious. And in exons, you can have both neutral, beneficial, and deleterious mutations. You can change this organization by using chromosome class in SLiM script. And also you can define the weight. So while you are running the simulation, you will define the genomic element. So you will say that simulate me a genomic element with composed of introns, exons, and UTRs, for example. And I want 90% of the mutations on the introns should be neutral and 10% are deleterious. You can change all these weights in your simulation code. There is also chance to do that. And the important thing is that if your study question is not about some parts of a chromosome, so if you're not asking about like the mutation rate between different parts of the chromosome, so the question is out of the chromosome, then you don't need to simulate the entire chromosome because it is more computationally demanding. Depending on your question, you should select. If you just need the genetic variation, if you're just studying the evolutionary forces and their dynamics, then you can just sequence like 10 megabase nucleotide long sequence or 5 kb nucleotide sequence long, but you don't need to produce an entire chromosome in your simulation to be just consistent. And also another concept is that the migration and subpopulations. So the subpopulations is that you create a population and then in each generation you can get another population from it. Normally the theme is that internally, so each subpopulation are internally panmictic but externally isolated. This is, for example, an example model with migration. This is a stepping stone model. You can also know about it from population genetics courses. In this model, each individual can only migrate between the close to two subpopulations. Here in this model, for example, you can simulate a scenario when there is a stepping stone model, simulate subpopulations like this, and migration rate from upstream to downstream is higher with the arrow here, and from downstream to upstream is lower. And in Wright-Fischer models, if you are simulating a Wright-Fischer model, as I said, like the constant population size, random mating, like the basic Wright-Fischer model, then your parents do not move between generations to mate with another individual. So the subpopulation here in the concept of the Wright-Fischer model-based simulation, the subpopulation is just a basic unit of reproductive isolation. And when you say population size change, the size of the population in the simulation changes in the next simulation, in the next generation. And in the Wright-Fischer models also, you should know that the population size never becomes 0. But you can do this in non-Wright-Fischer models. If you would like everyone to die, then you can simulate that scenario in non-Wright-Fischer models too. Another concept that we need to consider before writing the simulation script is the recombination. There are two ways to model the recombination in SLiM. One of them is crossover breakpoints model, and one of them is the double-stranded break model. I'm not going to cover this. This is a bit more complicated. But if you are interested in more complex evolutionary scenarios, like gene conversion, and you can just use the double-stranded break model to introduce the recombination, which is more biologically accurate. But in simple, while you are learning at the beginning, you can think that recombination can be modeled by crossover breakpoints model. Here, it assumes that there is the parental strands here, red one and the blue one, and we have some breakpoints. And then from those breakpoints, the chromosome is breakdown, and then this is the gametic genome. To do that, to give the rate of this recombination, you can just use initialized recombination rate method of the SLiM, and you can give this to your simulation, saying that yes, I want this recombination rate throughout my simulated sequence. And this is a bit faster compared to the double-stranded break model based recombination. So we say, Wright-Fischer models and non-Wright-Fischer models. At this point, I would like to also mention a little bit about the difference between these two. Normally, the Wright-Fischer model, you know probably is the basic model. And the SLiM's version 1 and 2 were supporting only Wright-Fisher model. In version 3, we also have a chance to simulate models in non-Wright-Fisher model too. To select a model while writing your script, as we saw in a couple of slides earlier, in your ADOS script, you can write initialize SLiM model type. You can use this method to select the model. So using this method, you can tell SLiM if you would like to select Wright-Fisher model or non-Wright-Fisher model. But if you are a very basic user, if you are starting for the first time trying to Wright-Fisher models and then becoming experienced and then proceeding to non-Wright-Fisher ones is preferred, because it's a bit more complicated. For example, in Wright-Fisher models, the generations are discrete. They do not overlap. So you have generation 1, and then they mate, and then you have the generation 2, 3, 4, and generation 100. And there is no concept of the age of the individual. Since one individual doesn't proceed to the generation 2, then there is no age concept. But in non-Wright-Fisher models, it's complicated but more flexible. For example, in the non-Wright-Fisher models, you can have generations that can be overlapping. And because of that, you have a age structure in the population, and you can have an age-related behavior in your population. Another thing is that in the Wright-Fisher model, if you are using it for the first time, it's nice because offsprings are generated automatically by SLiM, that considering the parameters that you provided. But in the non-Wright-Fisher models, you write a model, you provide in your script that model, and then individual dynamics of the model determines the offsprings that will be generated. Also, the population size is another thing that is different between Wright-Fisher and non-Wright-Fisher models. In the Wright-Fisher ones, the population size is controlled automatically by SLiM. Again, considering the parameters that you provided to the program. But in the non-Wright-Fisher models, the population size is an emergent property. So it means that it changes depending on the number of individuals die due to selection and number of individuals produced, and then this is more natural. So this is more natural. It depends on the carrying capacity of the population and fitness and density dependence. But on the other side, you should be aware about the script that you have written. And also fitness, probability of the individual will be apparent, affects the fecundity. In the non-Wright-Fisher models, however, the fitness is determined by the survival, and it is challenging. It is a bit challenging to model the fitness thing in non-Wright-Fisher models. So the thing is that if you start running simulations in SLiM, first start with the Wright-Fisher models, learn about the basics, about how to write the simple script, and then you can proceed to the non-Wright-Fisher models later. So the other thing that we need to know is that in SLiM, since we already saw that the mutations are not nucleotides when you are simulating, then they are just blue mutation, red mutation, you can imagine like that. But in nucleotide-based methods, it is also possible in SLiM version 3. You can start the model and generate the mutations. The nucleotide-based models can be simulated either in non-Wright-Fisher or Wright-Fisher way. And then nucleotides, you need to define nucleotide-based equals to true in your script. So in a couple of slides later, we will go through the scripts that you run in SLiM. Then this can be more apparent. So you just need to know that these are the methods that you write into your SLiM script. So the idea is that, maybe I can go back. Idea is that to run the simulation in SLiM, you need to have a TXT file that you have your script. And I am showing you about what are the methods that you use and what does it mean before writing your own script. So we will see them a bit more detailed later. In the nucleotide-based methods, if you would like to run a simulation to produce nucleotides, individuals with normal genomes. For example, you can simulate genomes and individuals, and then you can get the output in the form of the VCF files. And for that, you need to first have an ancestral nucleotide size. For that, you can write this method like initialize ancestral nucleotides method. You can do this by just giving a method like random nucleotides, or you can just load this ancestral nucleotide from a FASTA file. I will show you how to do that in the next slides. You can also define the mutations. It means that initialize mutation type nucleotide, you will say, and you can define the mutations as A, C, G, or T's, or you can define them as 0, 1, 2, 3. Here they correspond A, 1 is C, 2 is G, and 3 is T. And there is an option which says convert to substitution. This is important because at a couple of slides earlier, we saw that if a mutation reaches a fixation, then it is converted to substitution and removed from the simulation. But if your aim was to track that mutation, then you might not be asking SLiM to convert it to substitution. So you can change this default parameter here too. And the mutation rate is important. Normally, while we are writing a simple simulation scenario, which is not nucleotide based, in that one, we just give a mutation rate, a constant mutation rate that the SLiM introduces mutations randomly to the random positions. But if you are running a nucleotide based model, you don't need to, it's not very legal to use the mutation rate, overall mutation rate for generating nucleotide-based methods. Instead of that, you need to use a mutation matrix that gives a possibility of the mutation rate for each change. For example, if there is A to G mutation rate is like this, you need to have a four multiples for length matrix to give the rate of the mutations for each substitution. We will also see an example of these two. Basically, we are not going to be able to run the simulations together today because we don't have that much time. But I just wanted to give you an idea about how to install it and how to run it on the command line. So if you are a bit familiar with working on the command line, it will be just as simple as running SLiM and then parameter file. And to install, to download the source code, you can go to the web page of the SLiM. Maybe we can go together here. So here there are some source codes. There is an installer for the MacBook, and also here the manual, and also ADOS manual for the language. You can check this website. So maybe after the course, you can, if you would like to try, you can download it, and then after downloading, you will need some compilation of the program. I can share this with you. You can use this, but also in the manual, you can also find very detailed explanation about how to set up, how to install the program in your computer. After installing it, you just run it. Maybe I can show you just very simply. For example, this is my computer. So how do we run SLiM is that I downloaded it, I installed it. Now I just write SLiM, then it runs. This is the basic usage of the program. So it is like here you have the knowledge about the program, you have the web pages that you can follow. And then it also have a help part that tells you that write SLiM. And then if you want to run a simulation, put the script file here. So the usage is like you write SLiM here in your command line, and then you prepare a file that, for example, test.txt, and then since there is not the file, since I don't have this file here, then it will not run, but usage is that simple. You just write SLiM and the file, parameter file that you write all the script about the simulation. We will do this in the third class about how to write that script to run the simulation. So this is the usage. And the parameter file includes different things like mutation types, mutation rate again, and genomic element types, chromosome organization. You can define it in your parameter file. You can give the recombination rate. You can give the information about the generations. And also you can give the information about demography and structure. So output is optional. In some cases, you just run the simulation and compute the summary statistics. But in some cases, you can just generate the VCF files, like variant files, for example, for multiple individuals. You can put some parameters which are optional, like gene conversion or initialization, and the seed that defines that the program will be run from a user-defined seed. We can go through these basic examples and scenarios, and maybe we can try to. I can try to show you how to run such scenario in SLiM. And in the, how many times do we have? So we have just 5 minutes. So maybe I can just start with the neutral simulations, and we can continue with the other scenarios in the next classes. So this is a simple neutral scenario simulation. And what you do is like writing your script to tell SLiM to run the mutations. This is constant. You always start with writing initialize and open and close the parentheses. So maybe if I just put this here, and if I want to show you like this, how do you run the simulation? So we have the, maybe I can just, okay. So I have the program installed, and I will write a script. This is not a script actually. This is just a TXT file that tells SLiM to what to do. Let's open a text editor and open a file that we are going to write our scenario. So what we do is that at first, so in SLiM, if you read about the ADOS manual, you will see that normally while writing codes, you can be familiar, like you put this sign for just writing comments in R, but to write comments in your code, which means that you are not going to read it as code, but it will just a comment. Then you can use these two slashes, and here you can just say that set up a neutral simulation. And what I would like to show you is that what you do always while writing your simulation scenario, here you can just extend it. You can just add multiple comments here. So you can just write that I am writing my first SLiM code, such thing. This will be a neutral simulation. For example, you can write like this. So so how many times you want to write a comment, you can write it. There is not a limitation for this. And then you need to have an initialize part in your script. So it is like initialize. This means like you write initialize and then open and close the curly parentheses. Here in this part, what you are going to do is defining the initial parameters for your simulation. What you do is, for example, at first here, what we do is setting the overall mutation rate. This will not be a nucleotide-based simulation. This is just a simulation, simple neutral simulation. So maybe you can just copy from here. So you can write initialize mutation rate. This is the mutation rate. So you can change this. So if your mutation rate is different, then you can change it like this. So this is the mutation rate that you write here. And then what you do is like giving the initialize mutation type. So this is, you copy it, I paste it here. And then what do we do here is that my mutation type is mutation M1. You can just say it like A, B, C, X. It doesn't matter. I put its name as M1. And then this is 0.5 is the dominance coefficient. And then F is if the mutation will be reach to the, if the mutation will be reached to the fixation or not. And then the 0 here is the selection coefficient. Its order is always the same. So you need to write about the type of the mutation, and then dominance coefficient, the thing about the fixation, and then the selection coefficient. And after writing this, what you do is like, if you would like to, for example, sequence, if you would like to produce 100 kb long genomic sequence, then first you need to generate a genomic element type. This means that we first learned about like exons, introns, or UTR. So you need to say to SLiM that initialize a genomic element type, and this genomic element will be G1, and I will use mutation type M1 on this genomic element. And then you will say about the length of the genomic element that you would like to simulate. Here you say that I would like to simulate a sequence of 100 kb length, starting from 0 to 100 kb. You are writing here. And what you need is like writing the initialized recombination rate. So this is the recombination rate. This is just to initialize to start your simulation. This is not running the simulation. After starting, so you are defining the parameters here. You say to SLiM that I will simulate a sequence which is 100 kb long, and then this 100 kb long sequence will have some mutations, and I have in this simulation one mutation type which is M1, and then its selection coefficient and dominance coefficient is something like that. Then after initializing, what you do is to create a population. For example, let's say I will create a population of 500 individuals. Then what you do is like writing start, while starting write the generation 1. This means that it starts with the generation 1, and then open and close the curly parentheses again. Maybe you can follow if I write in this way better. And then open and close the parentheses. And what we do is SIM add subpopulation. So this is another method in SLiM to add the population. So if you write SIM add subpopulation P1, this is the name of my subpopulation, population P1, and number of individuals in this population will be 500. And start this population as 500 individual population, which has a mutation rate of this, recombination rate of this, and blah, blah, blah. And then what you do here is running the simulation. Maybe we can just come to here. And after starting your population, what you do is coming to the generation 10,000, for example. You started from generation 1. It is a forward time simulation. Start with generation 1 and come to the generation 10,000. And what you do is like in the generation 10,000, finish the simulation. If you just write this, let's try. Maybe we can also change the mutation rate as we to the original one. But it doesn't matter for now. So like we have the mutation rate here. So if I just say sim.simulation finished, then there won't be any output. Let's run it, SLiM, and then test.txt. Maybe I wrote something wrong. Maybe we can just do this like this. Okay. I did something wrong while writing the script, but normally when you're writing this, and if you say simulation finished, it doesn't generate an output. But if you say in the late event, so this is a late event, if you write 10,000 at the 10,000 generation, write me an output, then it will generate you an output with the mutations and all the information about the individuals. So probably we are run out of our time. So we can continue with other scenarios like nucleotide-based writing or the other ones after the break or in the third class. So I finish now.
There is a question, Gülşah, about is SLiM available on Docker?
I really don't know, but Maybe we can check together now, or I don't know.